Thanks to Joanne and thanks to the folks at Genome Atlantic for inviting me to speak to you today. Really, genetics is a major portion of many of the projects that we do in our laboratory, but I've tried to focus on a number of projects that I think really highlight the capacity of the zebrafish as a genetic model. I have one disclosure regarding one of the projects I'll talk about today with a patent pending for glycine and folic acid in our sideroblastic anemia project. And so really my goal today is to show you how the fish, which is a vertebrate but not a mammal, really relates to human disease. So I'm going to try and cover a fair bit today. I'm going to give you a couple slides of an overview of the zebrafish, why I think it's a good developmental and genetic model. I'm going to give you one example of some of the genetic manipulations we do for modeling cancers with our HACE1 story. I'm going to talk about how we use the zebrafish as part of the IGNITE projects with yeast models and other model systems to look at a couple of rare genetic diseases that have a preponderance here in the Maritimes. And then at the end, I just want to speak a little bit about our unique xenotransplantation platform that we've developed here in collaboration with Graham Dallaire's lab that we think really has a lot of potential as a preclinical drug discovery and drug response model. So first, why is the zebrafish a good model? Well, it's got highly conserved genetics. We usually say it's about 85% conserved with mammals, somewhat humbling since you can buy them at the pet shop for $2.50. Their development is very rapid, which I'll show you on the next slide. They produce large numbers of transparent embryos that are external, so that allows us, they're not in the womb, we're able to collect these embryos rapidly and manipulate them quite easily. Transgenic technology is very well developed. I'm not going to have a chance to really talk about transgenesis approaches today, but we have a number of transgenic projects in the laboratory. Transient gene knockdown using morpholinos and more permanent knockdown strategies using zinc fingers and talons and something new called CRISPRs are now really available in the zebrafish, and I'll touch on that today. With our xenograft project, show you how the ability to visualize the response of transplanted cancer cells in the fish in real time we think is a tremendous opportunity. So I'm going to show you this little movie. This is the only movie I have today. But this movie is really going to show you about the development of the zebrafish and give you an appreciation how quickly it develops and the optical clarity. So this is the zygote after it is split into two cells. And this is the yolk sac that provides nutrition for the first few days of life. Let's see if I can get this to play. And so now you'll see we're going through rapid development through these stages. We're now going through the blastula and gastrulation stage, where you have rapid cell division here over the first 10 hours or so of life of the embryo. Segmentation begins around now at 10 to 22 hours, and you're developing here the various different somites along the body of the zebrafish. This is inside profile. Here's the head. You can see the eye forming, the brain forming, the tail bud forming, and then this is a zebrafish embryo inside profile at about 22 hours post-fertilization. By 24 hours post-fertilization, the head and the tail are at right angles to each other, and circulation has begun. Blood cells are made anteriorly and posteriorly, and so you can see this is a very good model for studying blood development because things happen so quickly. So now I want to talk to you about this HACE1 tumor suppressor gene project, which we've been working along together with Paul Sorensen at UBC. This has been a cross-Canadian collaboration for longer than I was involved. When this was a patient that my colleague Conrad Fernandez looked after, it was a boy who had a Wilms tumor that was then found to have this abnormal translocation involving chromosomes 6 and 15, and through identifying this 6q21 region, this area, the HACE1 gene was found, and subsequently this was found to be an area of loss of heterozygosity in a number of tumors. Through work in Paul's lab, once HACE1 stands for the HEC domain and anchoring repeat containing E3 ubiquitin ligase protein 1, which is why we abbreviate it to HACE1. It has an anchoring repeat area, as mentioned, and a HEC domain, and is unique in having both of those domains. And as you can see here in human tissues, is conserved in a number of different tissues. As I mentioned, it's an E3 ubiquitin ligase, and for those of you not familiar, what an E3 ubiquitin ligase does is there are E1, E2, and E3 ligases, and their job is they function together as a complex to add ubiquitin groups onto substrate to target them for degradation by the proteasome, and the E3's job is really as the docking station. And so HACE1 functions in this way and is unique as the only ankyrin and HEC domain containing E3 ligase. So this protein was identified in human tumors and identified in some normal human tissues. Nobody really knew how it functioned to promote or to prevent oncogenesis when it's there and to cause oncogenesis in its absence, and nobody really knew what it did in normal development. And so Paul approached me as, could we use the zebrafish to do this type of work? You can see that HACE1 is conserved in zebrafish, and here using whole mount in situ hybridization, where we generate a RNA antisense oligo that binds to mRNA and shows expression. You can see expression. This is targeting that HEC domain. Here are zebrafish embryos at these different time points, hours post-fertilization, inside profile. So again, to orient you, here's the head and the body and the tail and the yolk sac. And you can see expression in a number of different regions indicated by the arrows in the brain here, in the heart, in the developing kidney. And then that's confirmed here by double whole mount in situ looking at colocalization at known markers for the brain, CROX20, for the kidney, CDH17, and for the heart, CMLC2, with those other markers shown in red and the HEC domain of HACE1 shown in blue. So similar expression to what we see in human tissues. So we wanted to see what happens from a developmental point of view. The zebrafish is a great developmental model. If it's expressed in the heart, is it important for heart development? So we took advantage of the CMLC2 EGFP fish. CMLC2 is cardiac myosin light chain 2, and so these fish have nice fluorescent green hearts. This transgenic line is provided to us by Ian Scott in Toronto. Normal zebrafish hearts have 2 chambers, not 4 chambers. Here you can nicely see the atrium and the ventricle. And interestingly, when we injected the HACE1 morpholino, you see what's called a looping defect, where you develop sort of this tube and abnormal heart structure. And this work was done by Lindsay McDonald, a former graduate student in the lab, and Babak, who's a current grad student in the lab and in the audience today. And he's gone on with other experiments to show that the heart rate and other functional abnormalities occur in these hearts. So through work that, so we're going to put that aside for a moment and think about what HACE1 does. And through work that Paul was doing in vitro in his lab, there was a suggestion that HACE1 may be playing a role in preventing ROS, or reactive oxygen species. And we know that reactive oxygen species are produced, and different reactive oxygen species include hydroxyl radicals, as well as hydrogen peroxide. And we know that in environments where you have a lot of reactive oxygen species, these can be pro-tumorigenic, creating that kind of environment. There's a number of different pathways that are involved in ROS generation, a mitochondrial pathway as well as an NADPH oxidase pathway shown here. It's quite complicated. There's a number of different components of these NOx enzymes, and this is an area of study I know near and dear to Karen Bedard in the audience. All of these different components, while this is shown in mammals, are conserved in zebrafish. And through work that Paul did both in vitro and then looking in mice using fluorescent labeling, and this is for hydroxyl radicals, he found that in various tissues in HACE1 knockout mice that there was increased levels of ROS illustrated here compared to wild type. And then Babak went on to show that this was actually the same in zebrafish. Here in the control, this is using a fluorescent pentafluorescein stain that labels specifically hydrogen peroxide, and you can see all these cells here are much more significantly increased in the HACE1 knockout fish compared to the control, and this is quantified using flow cytometry. In order to see which of these ROS pathways was involved, we thought it might be the NADPH oxidase pathway. Babak used a specific NADPH oxidase inhibitor called DPI. Each one of these images corresponds to one of these plots on the flow graph, and you can see here comparing the control to the DPI, HACE1 morpholino and HACE1 morpholino plus DPI, that we got a significant reduction, actually even lower than control levels in terms of reactive oxygen species, and this green line corresponds to this green curve here. And to further validate that, because that's what our reviewers wanted us to do, he used another NADPH oxidase inhibitor called apocyanin and saw a similar effect. So we thought that was all very cool and suggested that HACE1s may be playing a role in preventing cancer when it's present by controlling reactive oxygen species. And then we want to go back and say, well, does this play a role in normal development? And so we went back to that heart story I showed you where the HACE1 morpholino created this abnormal cardiac phenotype, this looping defect. And lo and behold, what we saw here was when we treated these embryos with N-acetylcysteine, which is a general raw scavenger, we were able to improve the cardiac phenotype back to normal, suggesting that not only is HACE1's role in controlling ROS important for oncogenesis, but maybe playing a role in normal cardiac development. Now, we were also interested in developing tumors in the fish, and so what we did here was the common mutation that's found in human HACE1 is this C876S mutation. It's a dominant negative mutation, and so what we did was we created transgenic fish overexpressing this human mutation. Some of them we just grew up, some of them we crossed with a P53 mutant fish because some of the tumors in mice, many of the tumors in mice had deletions of P53 to see if that would accelerate the phenotype, and these were all labeled with GFP so we could track these tumors, and found over time a number of fish that developed these masses shown here with GFP that are tumors. Here's another one developing something in the brain. And these are all, as mentioned before, HACE1 is expressed in the brain. And so we're currently histopathologically looking at these tumors and trying to see what their pathology is. Now, we generated these fish, as mentioned, using a dominant negative construct, and that's because while morpholinos, as I showed you, work very well in the short term in zebrafish embryos to look at knockout activity, they're really only transient. And so up until recently, we've had difficulties in the zebrafish compared to the mouse field being able to look at long-term knockouts because we're not able to do the classic homologous recombination experiments in zebrafish. That's been obviated with a number of technologies that have come online called zinc finger nucleases and talons. And this is a little schema here taken from this paper in Nature Protocols showing how the zinc fingers function by really as sort of modified primers binding on either side of the DNA, and they're connected to this protein called the FOC1 nuclease, which causes these double-stranded breaks, and then you get non-homologous end joining and get your insertion or deletion mutations. That's also shown here. And really talons function in a similar way, though they're even more modular because they don't have these three finger arrays, but are actually sort of single unit primers. And then work that William Lin, who is a undergraduate student in the lab working with Babat, he's now developed in collaboration with the Zebrafish Zinc Finger Consortium in Boston, has created a zinc finger that actually knocks out HACE1, and that's shown through the eye of faith a bit, this faint band here, where this restriction digest shows that he's able to remove HACE1, and those fish are now growing up in the lab, and I think will be very instructive in terms of telling us what happens with tumor development later on. Okay, so I'm now going to move on and talk about a couple of IGNITE projects and how funny-looking fish, as shown here in the cartoon, may inform us a bit about a number of these diseases. So the first story is one of cutis laxa. These are children that have abnormal skin, as shown here, and characteristic faces, and some of them have developmental delay. And this paper, which came out by another group in Nature Genetics, identified this gene called PYCR1 as a new cause of cutis laxa. There's a number of genetic causes of cutis laxa. This shows you the mutation here, and then this shows you the pathway that PYCR1 is involved in, the final step in making proline from glutamate. PYCR1 is the P5C reductase enzyme that's required. So this gene is conserved in zebrafish, and as Sergey, who's a postdoc in the lab, shows here, in the zebrafish genome, many genes are duplicated because that's what happened to teleos, those types of fish. They have duplication of the genome, so we have a PYCR1 and a PYCR2. And if you look here, that you can see expression in a number of different tissues. We would feel this is fairly ubiquitously expressed, but there's a fair bit of expression in the CNS and along the body of the embryo at these different time points. Similarly, PYCR2 is also expressed at these different time points, though it seems to be a little more restricted to parts of the brain. And so we knocked out both these genes together, PYCR1 and PYCR2, using a morpholino, and compared to the normal control embryos at 50 hours, again, this is inside profile, you can see that these fish are quite abnormal. You can actually even sort of appreciate that the skin here sort of looks a little bit wrinkly, and they're quite developmentally delayed. Interestingly, when we inject this morpholino in the p53 background, we somewhat rescue the phenotype, suggesting that this phenotype may be somewhat p53 dependent. And so, what's the cause of this phenotype? So Sergey went on to look at whether apoptosis is involved, whether these tissues are abnormal because the cells are dying prematurely. And so these embryos are all at 50 hours post-fertilization, and this is using anti-caspase antibody, caspase 3, which is the final step in apoptosis. You can see both bright field and fluorescent images here shown in red. Here's the head, here's the yolk, here's a portion of the body or the tail. This is baseline levels. In the double morphant embryos, you can see a significant increase in caspase staining, suggesting that there's apoptosis that's occurring. And then, interestingly, we're able to rescue a lot of that apoptosis when this is done in the p53 mutant embryos, suggesting that this process is p53 dependent. Similarly, using caspase 3 staining, Sergey looked at the CNS, and these are just 3 different fish. Here he's done the morpholinos separately, and you can see that there's a fair bit of apoptosis that's occurring in the CNS. And again, these patients have some developmental delays, so this phenotype may be consistent. So one would think that since this is an enzyme involved in the final step of proline metabolism, can we rescue this phenotype by treating with proline? And this was work that Gayat Nasrallah, who was a previous postdoc in the lab working on this project, did. He treated the embryos with proline, and he actually used 2 things as a readout. One was whether he was able to improve the morphology I showed you, and also survival, because many of these embryos, obviously, they're very abnormal looking. They did not live very long. The top figure here shows a time dependence of survival that was somewhat improved with the addition of proline, and shown down here a dose-dependent response that we saw, a nice dose-dependent response with regards to survival, and somewhat improvement with morphology. Now, interestingly, one of the wonderful advantages and opportunities of the IGNITE project is we're working with different investigators using different systems, and so a lot of the work on both this project and the sideroblastic anemia project I'm going to talk to you about next are done in conjunction with Chris McMaster's yeast lab. And this work was done by Pak Poon on this project, where he used the Pro3 knockout in yeast, which is the same as the PYCR1 gene, and showed that Pro3 knockout yeast are not particularly happy and do not survive very well compared to wild type. But as you increase the amount of proline, you're able to rescue that phenotype, showing conservation of what we were seeing in fish down to yeast. Now, so that suggests that proline may be part of the answer in terms of ameliorating the phenotype. But as I showed you both in yeast and also in fish, it didn't completely ameliorate the phenotype. And so one of the things that we then went on to do in yeast and in fish was to look at amino acid levels by HPLC and by mass spec. When we looked at those levels, we found that glutamate levels were actually down a little bit, but glutamine levels were quite high, as were ammonia levels in the morphins compared to the wild type. And not only were those levels high, but these enzymes, GLUA and GLUB, again, there's duplication of these enzymes. Both of those were quite a bit higher in the morphins compared to the wild type. And so one of the things that we wondered about was whether the issue here is not just the problem that there's low proline, because this enzyme indicated here with the blue arrow, the P5C, the PYCR1 enzyme, not only is there low proline, and so replacing the proline is the problem, but there's also increased levels of glutamine, whether we need to find ways to target other upstream enzymes to reduce glutamine in addition to replacement with proline. So we haven't done that yet. We're about to start looking at that. But again, this suggests how we can use the yeast in the fish to try to discern what's going on in these metabolic pathways. All right, so I'm going to move on to a second IGNITE project of sideroblastic anemia. Those of you that aren't familiar with this disease, it's a rare refractory anemia that occurs predominantly in children. And again, this project really was initiated by patients followed by Conrad in our clinic, who they developed these characteristic ring sideroblasts that have iron deposition here, shown in this image here, in this cartoon here, and this electron micrograph here. That's the pathognomonic feature that they've developed these ring sideroblasts. And classically, this has been an X-linked cause when it's hereditary, caused by mutations in ALAS2, and then more recently by the AMG group here, led by Dwayne Guernsey and Andrew Orr's work. This mutation, SLC25A38, was identified as an autosomal dominant cause. There's a number of acquired causes of sideroblastic anemia. So we want to characterize these a bit better. So first let me show you this pathway, which got us sort of thinking about what's going on. So here's the ALAS2 enzyme that catalyzes this step of glycine into ALA, and this is the cause of X-linked sideroblastic anemia. But you need glycine to come in. This represents the mitochondria, and you need glycine to come into the mitochondria in order to do this. And so the question was, is SLC25A38 actually a glycine transporter? And so if you have this mutation, never mind this conversion here, you actually don't even get glycine into the mitochondria. So again, the first thing we need to do is show that this was conserved in zebrafish. There's an A and a B gene of these in zebrafish, and again, here's in situs that Sergey did at different time points, showing that it's expressed, but actually showing that it's expressed in characteristic regions where hematopoiesis occurs. So in zebrafish, at this 24-hour time point, we see embryonic hematopoiesis here in the anterior lateral mesoderm and here in the posterior blood island, and so we actually see an abundance of stain here by in situ in those regions. And similarly in these other embryos at these later time points. And interestingly, we then took advantage of another transgenic line that we had in the lab. This is a GATA1 EGFP transgenic, which expresses green fluorescent protein in GATA1 positive cells, which are red cells. And then we're able to then look by doing PCR of these different populations, the fluorescent red cell population and the non-red cell population. You see here, these are hemoglobin genes that are increased in the red cell population. Interestingly, SLC25A is more specifically increased in the red cell population, in the red cell population, whereas A actually was higher in negative cells, so it's more ubiquitously expressed. But this at least confirmed to us that at least the B homolog was being expressed specifically in red cells. So then what we did is we wanted to see what happens when we take these double morphins and treat them with glycine. Again, with the thought being, if this is a glycine transporter, if we treat with glycine, can we ameliorate the phenotype? And so when we did that, these are zebrafish embryos now shown a dorsal view, so they're lying on their backs. You can see this is O-diadenosine staining, which labels hemoglobin over the yolk sac, and the morphins have basically completely eliminated, there's no staining there. This one, there's a bit of staining. They're completely anemic. And then when we treat with glycine, we're able to restore to some degree, not in all of our embryos, but a pretty good restoration of hemoglobinization. So we thought that was pretty cool. And again, this correlates with work done by Pedro and Noelia in Chris's lab in yeast. Here, the heme25A is the yeast mutant, and here you can see in terms of levels of hemoglobinization shown here, those are reduced, and those can be increased when glycine is added. They can also be increased when we treat with 5-ALA in yeast, which we didn't see conserved in zebrafish, which we think is important to what you see in yeast may not always translate into higher organisms, and important for us to move between those different systems. So if we quantify both, this is in fish, we're able to see that actually in the morpholinos we see a low amount of O-dianosine. We created an O-dianosine score for hemoglobinization from 1 to 5, and wild type sort of sits around here because there's variability, and in the morpholinos there's quite a low level, and then with glycine we're able to get in some, so this reflects sort of overall, because in some embryos we get a complete restoration, in some we get a partial, and so this sort of levels off at a sort of average score, if you will, of sort of close to 2.5 or 3, but it's not completely restoring wild type expression, and that's important. So this was also, so now we move back to yeast. So I'm going back and forth between yeast and fish, and this is on purpose because this is actually what we do on this project, where we learn things. And I'm not going to go through the entire history of how this developed, but another gene that's also involved in heme synthesis is this gene called GLY1. And so Pedro and Noelia looked in yeast and found that actually this is wild type hemoglobin expression, again shown here. Heme25, which is SLC, it's lower, but when you knock out GLY1 in yeast, that level is even lower than with Heme25, and both those levels are it's really quite low in the double knockout. And this is kind of interesting because the question was, if this is in the same pathway, so GLY1 is this threonine aldolase gene that's required to make threonine into glycine, and if this was in a linear pathway, it seemed odd in yeast that you would see a compound phenotype given that the same pathway is involved. And so, and that's really shown here as well, that there seems to be the synthetic genetic interaction between these two mutant yeasts. So again, here's wild type, here's the Heme25, here's the GLY1, but you really get very poor yeast growth with the double knockout. And this suggested that perhaps that there was a contribution of GLY1 in the synthesis, there was additional contribution of GLY1, not just in this linear pathway in the synthesis of heme. And so this cartoon shows you this complex pathway, and all I want you to take away from this is that in the mitochondrion, in addition to glycine being responsible for making heme, it also plays a role in making purines through generating thymidylate. And so one of the things that we wondered here is, given the importance of folate in this pathway, could the addition of folate further ameliorate this phenotype? And when, in yeast, when Pedro and Noelia added, so if you look, for example, here at this, this is the GLY1 and heme double mutant here, shown here in terms of growth, and if you added, these are all treated with glycine, and when you add folate as well, you increase the growth quite substantially. And when we did this in fish, what was exciting is that we similarly saw that effect. So if you remember before, we got a partial rescue in terms of our O-dianosine score with glycine alone, somewhat of a rescue, not great actually, just with folate alone, but then when we use both together, we almost get a complete rescue of hemoglobinization. So we thought this was really quite exciting, and there's work that we're doing together with a group in Boston who has a similar mouse model where we're looking at doing a clinical trial in these patients, and we think that our work informs the fact that in these patients, we may want to use both glycine and folate. It's interesting that this GLY gene in yeast and fish is a real gene. In humans, it's a pseudogene, so it's not expressed, suggesting that by us creating these knockout phenotypes in the yeast and the fish, we're replicating more what's happening in humans, and that using glycine alone may not be curative in humans, but the addition of folate may really improve glycine as a therapy. So we think that's very exciting, and as I mentioned, we've now submitted a patent for this combination therapy in this disease. So for the last bit of my talk, I'd like to talk a little bit about our xenotransplantation work. So we're switching gears. So this is to remind you we're doing something different. And for those of you that can't read this, this fish says, "Oh, now the old bowl looks pretty good. Didn't I tell you the water's always bluer on the other side of the glass?" So this is a different approach to using zebrafish. And this work really derived from this thought that we wanted to develop a quantitative cell proliferation assay in vivo to look at the response of human cancer cells to anti-cancer agents in an environment that we thought was more like what's seen in humans. So a lot of work is done in cell culture, and obviously that's been very informative, but it really lacks the context of the microenvironment, which we know makes a major contribution to how cancer cells are going to respond to treatments. And so this work, now published a couple of years ago, led by Dale Corkery, who's in the audience today, really allowed us to take the fish and develop this platform for drug responses. So this is a little cartoon showing the protocol that we use. We take human cells, human cancer cells. These are unlabeled cells. We fluorescently label them with this dye called CMDII and collect those cells. Those cells are then injected into zebrafish embryos at 48 hours. At 48 hours, zebrafish embryos do not have an adaptive immune response, so we don't need any immunosuppression in order to do this. We screen these embryos for fluorescence, and then we're able to monitor and follow those embryos. This is shown here instead of cartoons using real pictures. So these are the fluorescently labeled cells that we've gotten from culture. The fish that we use for these are these so-called Casper double pigment mutants that Rich White made in Len Zon's lab in Boston. They're a cross between the Roy Orbison mutant, I don't have to tell you why it's called that, and the Nacre mutant. And these fish are really a zebrafish that have lost their stripes. They're completely transparent into adulthood. People like to say you can read the newspaper through these fish. You can see the heart and the eggs here and all the organs. And so we use these in our xenotransplantation because the optical clarity is really improved. We inject somewhere between 50 and 200 cells into the yolk sac and then screen fluorescent mass, and this would be a positive fish. And in our lab, we've now done this with quite a number of cell lines. This is an old slide already. This is showing a number of breast cancer cell lines on the top and a number of leukemia cell lines on the bottom with bright field images and fluorescent images. Again, these fish are in side profile. HPI is hours post-injection. And so you can appreciate, I think, in all of these cases, in the fluorescent images better, that this bolus of cells that's injected enlarges and those cells are proliferating. Now, while that's been very helpful to be able to look at it by live imaging, we wanted to create a system where we could actually quantify this, because in order to be able to use this for drug discovery, just looking and saying, well, the numbers look increased or decreased, we thought really wasn't going to be particularly robust. And so in order to do that, we developed this in silico cell proliferation assay where in a given time period, this is showing you 24 hours and 72 hours, the fish are dissociated into a single cell suspension. We pull out these cells, we put them on a microscope, and then using a grid and the ImageJ program from the NIH, we look at the number of cells at these different time points, and that tells us the increase in cell number. And we can similarly apply this assay to look at drug responses, as you'll see. So we then took on this proof of principle study to see if this works. This is using two leukemia cell lines, K562 cells, which is a CML leukemia cell line, and NB4, which is a promyelocytic cell line, and the relevance of those cell lines will become clear in a moment. And when we xenotransplanted those cells into the fish, using that assay I showed you, Dale found that there was a significant increase in proliferation here at 24 hours compared to 72 hours for each of those cell lines. And importantly, that proliferation paralleled what's seen in vitro. Similar kinetics for both the NB4 and the K562 cell lines. You'll notice here in vitro that the proliferation is a bit faster than in vivo, which isn't surprising given the fact that in vitro all the key cytokines, etc., are added to the media to enhance growth. Now, the reason we chose these specific cell lines initially is because each of these cell lines harbors a specific targetable mutation. So in the K562 cells, they have the BCR-ABL mutation found in CML that's targeted by imatinib mesylate and other tyrosine kinase inhibitors, whereas in the NB4 cells that are in acute promyelocytic leukemia, They harbor the PML-RAR alpha, which is found in APL and targeted by Atron. Both of these drugs are used commonplace as the standard of care for these diseases currently. And so what we found was when we xenographed those cells into fish and then added these drugs to the water, we saw that in the NB4 cells, by adding ATRA compared to vehicle control, we could see a significant decrease in cell number by proliferation, and similarly using imatinib in the K562 cells. And importantly, this was a key experiment, the reciprocal experiment, that when we added imatinib to these cells, to these fish, or ATRA to these fish, we didn't see the same kind of response, suggesting we were really seeing a targeted therapeutic response and not just general toxicity of this drug on the cells. And I'd mention, because we're often asked this, the fish look totally fine through this process. So based on that work, we did a collaboration with Alison Thompson in the Department of Chemistry at Dal, where she had been making derivatives of a natural compound called prodigiocin, which had been used to treat leukemia many years ago, but was found to be too toxic. And so she was developing these prodigiocin derivatives, and had been testing these for efficacy in a number of cancer cell lines from the NIH, including anti-leukemia cell lines. This work that I'm going to talk about was recently published and was a cover of this journal, Organic and Biomolecular Chemistry. So Alison and Deb Smith in her lab did all this fancy stuff that I don't know what exactly it is, but she generated these various different derivatives, and they're each labeled DSB and then a number. And when she used these different drugs on NCI panel cell lines, she found actually that the leukemia cell lines were the most sensitive, and those are shown here for each of these different compounds and indicated by the name of the cells and this symbol. You'll notice one of the cells of the K562 cells that we had used in fish, and so based on that and the fact that those K562 cells, we know we had a positive control with imatinib, we wanted to see if these same responses were seen in vivo. And so what we did was, as we had done with imatinib, we xenografted the K562 cells into zebrafish. Here's our imatinib positive control. Here's untreated just with DMSO. And like the imatinib, this particular compound, DSB8, at this dose, we saw significant inhibition and proliferation, suggesting for the first time that you could use the zebrafish xenograft assay as a way to look at responses for novel drugs that are being synthesized or compounds in drug libraries. So the nice thing about doing fish work is there's lots of great cartoons you can find. So how can we use this for treatment? So this cartoon, if you can't read it, this fish says to this fish, with all these omega-3 fatty acids, you think I'd feel better. So how can we use this for personalized therapy? Because as many of you know, I think in oncology, in lots of fields, but particularly in oncology, there's this goal towards personalized therapy, or sometimes called precision therapy, where we're trying to, based on genomics, really drill down and treat a particular profile of disease for that individual, rather than sort of treating all comers with a more one-size-fits-all. And so this was sort of initially a dream slide. This used to be one of the last slides of my presentation, and now I'm proud to say we've been able to actually make it a reality. So this is a child, let's say, who has leukemia. We do bone marrows for diagnosis, and here we take some of that bone marrow sample and fluorescently label it, put it into the zebrafish in the xenograft platform that I showed you, treat with different drugs as I've shown you for the cell lines, and then separate those out and quantify and look for responses. And we have already approval from the IWK to do this, and over the last year or so have, I think we're up to almost 30 samples now of children with different leukemias. We've only had one family refuse. So this shows you our process. We take these cells and we separate them out. Actually, we've been able to do this, as I'll show you, with both whole bone marrow as well as buffy coat. We culture these cells, we freeze some down, and we're able to do it both from fresh cells and from frozen cells, and we're able to then do injections on day 2. And you'll see here that we get engraftment by day 4, and we're able to look at proliferation by day 7. So this kind of assay, to be able to do xenografts of primary human samples and then test drugs, is completely unheard of in the mouse. Many of primary samples won't engraft in the mouse. Of course, you're using complex immunodeficient mice, and there's a whole discussion about whether those immunodeficient mice, how much they recapitulate what's going on in a human, and depending on how immunosuppressed that mouse is, you're going to see different responses. And even if you get that all to work, it's going to take you 3 months minimum in a mouse, and we're able to do this in a week. And so we inject these into the fish, and then we get these readouts. So here's a couple of examples. Here's a child that had pre-ALL, which is the most common type of leukemia, the most common cancer we see in children. If you look here at 48 hours compared to 144 hours, this is at 5x magnification and 10x magnification. You can appreciate that we see proliferation, similar to what we've seen for the cell lines. This is a patient with TALL, which is a disease that, as you'll see, we're quite interested in because these patients don't have the same type of outcome as those with pre-BALL, and we really need novel therapies. And so here again, we're able to see engraftment between 24 and 96 hours, which shows up better here at higher magnification. So T-cell ALL is a very interesting disease. We've made some progress, some headway in these patients, but whereas for all comers really with pre-BALL, we have a cure rate sort of in the 90% range, we're nowhere near that with TALL. We're much more in the 60 to 70% range. What we do know about TALL is we know about a lot of pathways. We know about the PI3 kinase pathways involved. We know that the AKT pathway is involved, the mTOR pathway is involved, and the Notch pathway is involved. And we have a number of different drugs, rapamycin, which targets mTOR, tricerabine, which targets AKT, and compound E, which targets the Notch pathway, which have been developed. Some of these have been used with some success, varying degrees of success in clinical trials, but target various aspects of this pathway. And so we wanted to know, could we take advantage of our xenograft model to look at these pathways? So here's an example. Here's another patient who had primary TALL. We engrafted his cells into the embryo. They proliferate. This is with vehicle over time. We tried treating with rapamycin, and we've done dose-response curves for all of these doses. We don't really see much response. We try, but then when we treat with, and we did the same thing with tricerabine, but then when we treat with compound E, for instance, we're able to see that that bolus really of cells really doesn't divide and starts dividing up, suggesting that this may be efficacious. Compound E targets the Notch pathway, and then through work together with Karen Bedard and folks in her lab, we went and found that this patient actually had a novel mutation affecting Notch1. So here, not only were we able to use the fish to find, to look for a drug response and identify a targeted therapy that might work, but we were able to, within the span of a couple of days, go back and actually use drug response to identify genotype. So here we have genotype, phenotype, and drug correlation, all from this in vivo assay in fish. And based on these results, last year, Graham and I, together with a group in Toronto, got funding from CIHR and from NSERC through a collaborative health research project grant with GSK to look at novel inhibitors in some of these pathways in TALL. Yeah, so there's the mutation there. So putting this all together from a drug discovery point of view, we think that the fish has a lot of potential. And as thinking about this in some ways, the way we've thought about the way the IGNITE project has worked as a pipeline, we can use fish that we've, as I've shown you with HACE1, that we've used, that either have, we've used morpholinos or knockout strategies. I haven't shown you today, but we have a number of other projects in the lab where we've got transgenic models of a number of diseases where we have embryonic readouts. These fish, because they're tiny, can be arrayed even in something as small as a 96-well PCR plate with a number of embryos per well. We can treat them with different drugs. We can then look at those fish that are developmentally normal and compare drug responses and look at a readout, which may be using in situ hybridization, as I've shown you, looking at a restoration of a normal phenotype. We can use fluorescent, as I've shown you, we have a number of fluorescent lines and may be able to use that as a readout. Then together with labs like Chris McMaster's, we can then look at what that compound was and try to optimize that compound. We can then take that into cell lines together with labs like Karen Bedard's, going back into human cell lines and seeing if there's that response. We can then take those human cell lines into the xenograft model we've developed and see if what we're seeing in vitro translates in vivo. And it's not shown here, but obviously, like the TALL project, we can then go to mouse collaborators and see if our best hits that we've screened through in the fish can then translate into mice and other preclinical models. And then ultimately, the goal, of course, is to take these to the clinic. And with my other hat on as a clinician and being involved with clinical trials through groups like the Children's Oncology Group, we can help sort of facilitate the movement of these drugs from the lab to the bedside. And I can tell you from being at a number of recent clinical trials meetings, both in children and adults, there's a real appetite to try to do this in these diseases and try to bring these new drugs into the clinic a bit faster than we have been in the past. So in summary, I've talked about a lot of things today. I'll try and summarize it just in these couple of points. The zebrafish, I hope I've convinced you, is a useful in vivo tool for investigating mechanisms underlying oncogene and tumor suppressor genes. Again, I focused on this story we have with HACE1 as a tumor suppressor gene, but through other work in the lab, Adam DeVoe, who's a graduate student who's also in the audience today, is working on a high-risk leukemia model that's based on a transgenic strategy. Similarly, we've been able to identify mechanisms underlying malignant progression. That using knockdown and transgenic technology in the fish, we can create a rapid in vivo readout for genotype-phenotype correlations and responses to drugs. And then the last bit of the talk, I hope I've been able to show you that this xenograft platform that we've developed really, I think, has a lot of potential in a pipeline, both to provide preclinical information and potentially help personalize our management of children and adults with leukemia and solid tumors. So I've been fortunate enough, this is a very busy slide, but this, you never want to have busy slides, but I'm always proud to have a busy acknowledgement slide because it means that we work with lots of great people. This is the lab currently. I've highlighted in bold text those people in the lab that have been involved with the work I've shown you, and other people in the lab have been working on other things, and I think I mentioned them sort of as we went along. A lot of the work on the HACE1 project was done by Lindsay McDonald, and now has been led by Babak with help from William. And the Ignite work was done by Gayev, now being taken over by Sergey and Shelby Steele, who's a new postdoc who's joined the lab, who's working on a number of other projects that are in early stages I didn't have time to talk about today. And then the xenograph work has been really championed by Dale Corkery, and now has had Victoria and Chansey, who's actually done a lot of work on solid tumors in the xenograph model that I didn't have time to talk about today, and have really moved this forward. And now Vino Kumar and Sergey in my lab have also joined the Xeno team. In terms of collaborators, I've mentioned Graham and Allison for some of the Xeno work. All the Xeno work has been done in collaboration with Graham, who has provided a lot of expertise in terms of cell culture and imaging, Allison for the Prodigiosin project, and then collaborations, as I mentioned, on HACE1 with Paul Sorensen, and on the T-cell project with Andy Wang, both at UBC. And then, of course, our collaborators on the Ignite project, who I've mentioned, those folks in Chris's lab who've done the East work, as well as a number of other people on the Ignite project, and our funding. So thanks very much. I'll be happy to take questions. Speechless.
Just curious.
Oh.
Sorry, Joe.
Okay, go ahead.
The derivatives that you mentioned, the chemistry person, are they in the public domain, or are they new things that are maybe going to be patented as well?
So they're new things. They're not in the public domain as yet. This paper came out in January, and I know that Allison's working to further improve these derivatives, and then ultimately for us to try to take them into some mouse models. Chris?
When after you've xenografted your fish and plus or minus drug, are you thinking about maybe doing, again, some exome sequencing or something like that to see if they required any mutations?
Right, so one of the... Great, so that's a great question. So one of the real nice things I like about the xenograft platform is, as I sort of alluded to in a couple places in my talk, the zebrafish, in terms of some of the technologies we have available, is a bit behind some other model systems. It's a newer system. The first paper in xenografts came out in 2005, so this is a really new field. And so a lot of the technologies that we might have available for fish are a bit behind. The nice thing with the xenograft is it's human cell lines, and so anything that could be done in human cell lines, either in a petri dish or passage through a mouse, we should be able to do through a fish. And so that'd be very interesting for us to look at pulling those cells out after treatments and looking at by microarray or RNA-seq, or looking at what happens in terms of acquired mutations and responses to treatment. Of course, we can then do the flip side and pull out components, pull out the fish or components of the fish, and look at those and see how the microenvironment may change in response to those drugs. What I didn't really have time to show today is one of the other advantages we think with the xenograft platform is, given the transparency and the optical clarity of the fish, we can xenograft these cells into a number of fish lines where we have fluorescently labeled components of the microenvironment, so the vasculature, various immune cells, and so we can then directly look at those interactions in response to drugs in a way that I think would be challenging to do in a mouse system. Conrad.
Great talk, Jason. The question I had was, when we think about treatment for cancer patients, it's a longitudinal treatment with many different drugs over time, and in a mouse model, for example, you might have an opportunity to look at that over a long period of time. Can you talk about how that might parallel things in the zebrafish?
Right, so that's a good question, because most of the assays that we've done here in the embryos have been are short-lived in terms of responses. So, the advantage to that sort of short time interval is, can we look at a response and use that as a window in real time to then go back in a clinical trial and say, well, we've tested this panel of drugs in your tumor in a petri dish and in a fish, and so we think that you have more response to this tumor, your tumor will have more response to this drug. So I think that's an advantage. For longitudinal studies, I think you're right, this xenograft model may not hold up. We've had these fish live about as long as 2.5 weeks, and then when they're not treated, they get overcome with tumor and they don't do that well, and then we sacrifice them. In theory, in fish, we could do xenografts in adult fish. People have done that. And in fact, Vino, in our lab, in the lab that he did his undergraduate in, they did some of that. I think that one could look at, just like we would potentially take these drugs into mice for those longer-term studies, we could look in adult fish and use that as a parallel approach. So at this point, I sort of see this really more as a drug discovery tool. I think we've been asked quite a bit also, can you use the fish to look at, various toxicities to drugs? And we are starting to do that. We have a collaboration with Randy Peterson's lab looking at drugs that prevent anthracycline toxicity and looking at that in fish. I think some of that is going to be possible, but I think ultimately, the purpose of our work is not to exclude the mouse, but how can we use this to better use the mouse in a more cost-effective way, and perhaps then go to mouse and other models to look at drug responses, long-term effects, side effects.
You might be able to re-inject some of the treated patients and see how they respond, or the fish respond differently to this.
Yeah, doing sequential dosing. Yeah.
See what happens over time.
Yeah.
Just a question from the fish perspective. If you're putting in all these drugs and seeing an effect on the fluorescent cells, I guess the question is, from the fish perspective, how do you know that there isn't changes, or what are the controls on the fish side that the drugs aren't affecting those and therefore affecting the effects that you see on the fluorescent cells? Right,
so I think that's a great question. I guess you could answer that in a couple of ways. One could say, this is more in the philosophical realm, that if you're treating a mouse or you're treating a person, you're also not just treating the tumor cells, you're treating the whole organism. And so, again, treating these transplanted cells in the fish is that more representative? I think through some of the things like Chris was talking about, looking at both in the host and in the cells, changes after the drug, either by genetic profiling or by other means, may be able to get at those differences. Developmentally, the fish look normal and look well. But are we affecting other pathways? I mean, that may be good or bad. You're right, and I think those are things as we move ahead with this assay that we need to take into consideration.
Jason, also, a second question is, we often use drugs in combination. Have you tried that yet in a fish in terms of complementary chemotherapy agents?
Right, so this is good because then, Dale and Victoria know what they're going to be working on next week. So, so certainly we can use this approach to do that. The other thing, and we've started looking at, there's no reason why we can't use combination drugs, and I think that's also going to be important because, as you well know, a lot of these new agents, they're not going to be, no one's going to move them into the clinic on their own. They're going to be, you may do a phase 1 trial, but, especially if it's an agent that we know, we want to see how does that potentially enhance current therapy, or it's going to be added to a backbone of standard therapy. So I think we should be able to do those studies in the fish. Adding additional drugs to the fish I don't think is tricky. We've also looked at, people have asked us about doing competitive cell populations and looking at putting more than one cell line in and what happens. And those are a little trickier. But I think the drug studies are ones that combination therapy should be feasible, at least for short-term responses.
Thank you, Jason.
Thank you. Thanks very much.